Welcome to The Explainer. Today, we're diving into what is honestly the ultimate biological logic puzzle, genetic mapping. We're going to literally decode the invisible architecture of life itself. I mean, really think about it. How do we know exactly where a gene lives on a chromosome? If genes sit on chromosomes kind of like stops on a train line, how do we actually map the tracks when they're way too tiny to see with a regular microscope? It's not like we can just pull out a teeny tiny ruler, right?
So here's our roadmap for this explainer. We'll look at what genetic mapping actually is, the mechanics of crossing over and recombinance, how to calculate recombination frequency, measuring distance in map units, and finally, sequencing genes on chromosomes. Let's get into it.
All right, starting off with part 1. What is genetic mapping?
By definition, a genetic map is basically an ordered list of genetic loci along a particular chromosome. We use it to study their relative position and sequence, but here's the catch. We can only build this map for linked genes, meaning genes that live on the exact same chromosome. And the whole thing entirely depends on a natural biological phenomenon that happens during meiosis, which is called crossing over.
Which brings us perfectly to section 2, crossing over and recombinance. Okay, so the golden rule of physical distance is this. Genes that are super close to each other, they rarely separate. They can't easily be broken apart by crossing over during meiosis, so they're usually inherited together. But genes that are far apart, they have a way higher probability that a crossing over event will sneak right in between them. Going back to our train analogy, close neighboring towns almost never have a massive detour drop right in between them. But for towns sitting way far apart on the line, there's a much higher chance the tracks might switch. And when those tracks do switch during crossing over, we get completely new combinations of genes. Offspring that inherit these brand new combinations, ones that look different from their parents, are called recombinants. And honestly, this brilliantly illustrates the foundation of our math today. The exact proportion of these unique recombinant offspring in the total pool is literally the secret to measuring the physical distance between genes. So how do we find that precise proportion? That is section 3, calculating recombination frequency. Time to introduce our absolute best tool as genetic detectives, the math formula. Recombination frequency, sometimes called the crossover value, is equal to the total number of recombinant offspring divided by the total number of offspring, all multiplied by 100. Why does this matter? Well, this formula is exactly how we translate the messy, unpredictable reality of biological reproduction into clean, measurable percentages. Let's look at some actual data from a fruit fly test cross from the source material. Check out the parental types. These are the traits passed down together. We have gray body with normal wings sitting at 237 and black body with vestigial wings at 241. Notice how these parental types absolutely dominate the total number. But look at the bottom 2 rows. Gray body with vestigial wings at 44 and black body with normal wings at 48. These new mixed and matched combos, those are our recombinants. They're the smaller numbers for sure, but they hold the absolute key to our map. Let's actually put the formula to work. We add our recombinants together, so 44 plus 48, which gives us 92. Then we divide that 92 by the total number of all offspring. That's 237 plus 241 plus 44 plus 48, which gives us exactly 570. Divide 92 by 570, multiply by 100, and boom, we get exactly 16.14%. That is our recombination frequency, calculated straight from physical breeding data. Now we have a solid percentage, but we need to turn that back into a physical map. On to section 4, measuring distance in map units. So the absolutely crucial point is this golden rule. Distances between genes are expressed in map units, and one map unit is equivalent to exactly a 1% recombination frequency. By the way, a map unit is also formally known as a centimorgan. This direct, one-to-one conversion is the whole reason genetic mapping is even possible. Applying that to our fruit fly data, our 16.14% recombination frequency translates directly, and I mean literally 1-to-1, to 16.14 map units or centimorgans. So we can wrap up this specific example by confidently stating that the gene for fruit fly body color and the gene for wing shape sit exactly 16.14 map units apart from each other on their chromosome track. Which brings us to the grand finale, section 5, sequencing genes on chromosomes. To actually solve these sequence puzzles, we use a foolproof, highly logical strategy. Step 1. Find the highest crossing over value in your data. This represents the farthest genes. Step 2, place those genes at opposite ends of your map to establish your outer boundaries. And step 3, use the smaller crossover values to place the remaining genes right in between them. Let's see how this builds using some raw data for 4 mysterious linked genes, P, Q, R, and S. We've got their crossing over frequencies here, P and Q at 35%, P and R at 5%, R and Q at 40%, Q and S at 10%, and R and S at 30%. I know, it looks like a total jumble of numbers right now, but stick with me and just follow the steps. Applying step 1, we scan for the largest number. R and Q clearly have the highest crossing over value at 40%. Since 1% is 1 map unit, that means they're exactly 40 map units apart. So we drop R and Q at the extreme ends of our map. Just like that, our outer boundaries are completely locked in. Now we work through the next biggest clues to slot in our inside pieces. We need to place gene P. The data says P and R are 5 map units apart, and P and Q are 35 map units apart. Well, if we place P just 5 map units to the right of R, it perfectly leaves the required 35 units between P and Q, fitting right inside our 40-unit boundary. It's basically a giant puzzle. Now watch how our final gene, S, slots perfectly into place. The data states S is 10 map units from Q and 30 map units from R. So, if we drop S 10 units to the left of Q, it exactly matches the 30-unit distance back to R. And just like that, the puzzle is solved. We have our final relative sequence. R, then P, then S, then Q. The real beauty of this system is that the exact same logic applies no matter how many genes you throw at it. Let's briefly look at this larger 5-gene puzzle from the source material involving genes J, K, L, M, and N. It seems pretty daunting with all these percentages, but remember your rules. You always start with the largest value. Here, it's J and N at 70%. You set those as your anchors at 70 map units apart, and then you just work your way inward by filling in the remaining distances step by step. So M sits 20 units from J, L sits 15 units from M, and K sits exactly 15 units from L and 50 from M. We reveal the final solution, JMLKN. This just proves that our mathematical detective method works every single time to reliably plot the physical layout of the chromosome. We're turning abstract breeding data into a totally concrete biological map. We've gone from simply counting fruit fly offspring to literally drawing a map of invisible DNA. Which leaves us with a fascinating final thought for this explainer. With these incredibly precise mathematical tools, what other hidden genetic maps can we decode across the natural world? What other mysteries of inheritance are just waiting for us to calculate the distance?